All right. Well, then, welcome everyone. Thanks for joining for today's um, iteration of the seminar series, which is very promising and will be great, I promise. So I'm happy to welcome Aurélien Roux to join. Aurélien is a trained biologist and physicist. He got his PhD in 2004 at the Institute Curie, and then did a short postdoc at Yale, and then he started his independent career as a first researcher, CNRS researcher at the Institute Curie again, before becoming an assistant professor at the University of Geneva, where he rose through the ranks. Also, he's there at the Department of Biochemistry, and in 2019 he became an elected EMBO member. And in Aurelian's lab, they're very interested in membrane properties and everything related to membranes, including membrane trafficking, cell division, and cell migration. And this is, I think, the topic that he has been developing also over the last couple of years. And we will hear today about cell forces, order and flows during in vitro reconstitution morphogenesis. So thanks a lot for being here, and the floor is yours.
Thanks, Gabriel. And indeed, what I will be talking about today is something new uh, for the lab. I mean, new. Um, it has been quite some years we've been working on, but the first papers we've published were published since 2020. So it's a pretty recent aspect. So today I'm gonna get inspiration from things that have been found in the cytoplasm, so cytoskeletal filaments, and I'll show you how through a soft matter approach you can develop a body of theories that can understand also the morphogenesis of higher scales, so tissue scales instead of cytoplasmic aspect. And this will be essentially the only link, I would say, to cytoplasm, since the main focus of this seminar series is the cytoplasm. So what we look at is embryonic development, and in particular, we would like to reproduce in the test tube, so in vitro, different steps of morphogenesis. And what you see during this beautiful movie of the Drosophila embryo development is essentially three processes that are coupled together, that they work together. So the first thing, of course, which is linked to the term morphogenesis, is shape changes of the tissue. But you also see flows, and in particular in that movie at the beginning, when gastrulation occurs, you really see that there's a lot of movement of cells. And now you see it again when you have the closure of the tissue that is happening, and you see the cells moving in and things are moving around. And associated with these shape changes and flows, you also have order. You can see that sometimes the cells are elongated. In this case, these are nuclei that you're looking at gets elongated, and these elongation can create some sort of orders. You also see the segmentation of the embryo that is very clear on this movie. So the combination of how you remodel the tissue, how you flow the cells, so the fluidic aspects or hydrofluidic aspects of the tissue is important in conjunction with the order of the cells. So these are characteristics of a bunch of materials that are called active materials. And in particular, there has been a body of work on trying to reconstitute those active systems that you find in cells in vitro. And one of the most famous one is the one from Zvonimir Dogic, and is shown here, where they created a 2D nematic of active cytoskeleton. And the way this was done by essentially taking stable microtubules and mixing them with kinesin motor clusters, which actually have two kinesin motors bound to streptavidin, and that allows to make cross-linking kinesin motors, which will bridge different microtubules, as you see in the drawing here. Why is that important? Is because now you have a system that will condensate the cytoskeletal filaments into a thin layer at the surface of oil and at the interface, I should say, between oil and water. And this 2D layer with highly condensate of microtubules will form a nematic order. The filaments will essentially align locally because of density. They get closer and they are elongated molecules, so they will just align locally. And because of the action of motors, not only they align, but they form this beautiful nematic order that moves with time, creating flows and creating defects. And I'm going to come back on this in a second. But you see that in vitro, this is a 2D system. You can reproduce a sort of similar thing than what I showed you in the previous movie, which is a mix of flows, order, and deformation. So using the framework of liquid crystal phases and liquid crystal description is interesting at different levels for biology. The first level is that there's a ton of different types of liquid crystal phases. These are the most famous ones, but also there's a lot of different behavior, in particular when they're subjected to flows or to phase transitions, meaning that you change the temperature of the system or you put energy in the system. The one we'll be talking about today will be mostly the nematic phase, where essentially you have elongated particles, and because of a high density, they will locally align and give a global order. All the molecules will be globally ordered in the same direction, essentially. And that's what we see with this system of microtubules and kinesins on the 2D, is that locally you have all the microtubules are oriented the same way. Nevertheless, you see that there are curves, and in between the curves, you see defects, and defects are points where you lose the order, where the microtubules are not perfectly aligned, or even disappear, as you can see from the black spot that you see sometimes. In those active systems, you actually have two kinds of defects, the plus 1/2 and the minus 1/2. So these are the ones you see. We call it the half-integer defects. And I'll show you that the number that we give plus 1/2 or minus 1/2, which is called the charge, is actually a topological description of this defect and is related to the pattern of orientation around the defect. And I'm going to show you this in a second. So the half integers that you see here are interesting because, as you may have seen from the movie, the plus 1/2 are moving while the minus 1/2 are essentially static. And the plus 1/2 are moving because, as you can see from the shape of the microtubules around the defect, they are asymmetric, which means that if you integrate the forces around the defect, you have a net flux or a net force that pushes the defect toward a certain direction. Now you have other types of defects which cost more energy to create. These are the integer defect. And these defects, as you can see, are completely isotropic, meaning that the orientation field is the same in every direction, with the exception of the -1 defect. And so the isotropic defect of which are integer, or another class of defect if you distinguish them from the half integer defect. So active system essentially make on the flat surface plus and minus half defects. Now, what happens if you put them on a curved substrate? And this you can do using liposomes, which is one of the favorite tools we had in the lab. So this is not our experiment. These are experiments done by Andreas Bausch. And what they did was to take the same system that I showed you before, kinesins and microtubules, and through a gluing system, put them onto the liposomes. And in that case, you have a topological constraint because the liposome is a sphere. So that means it's a closed surface with a certain curvature, and that will impose a certain organization to the filaments. They cannot be fully ordered in every direction. So what they do is to actually, in some cases, organize as concentric circles at the equator plane of the liposome. And you see the picture below. And so that means essentially that it creates two integer defects, which are the poles of the liposome, and it spins around. So it's maybe you don't see it very well from the picture below, but you see that there's time frames, and in those time frames, you see that the movie or the configuration of the microtubule is changing. In that case, you don't really see any deformation of the liposome. Liposomes are very soft material because they are made of lipid membranes, and these are very, very easy to deform. But if you get another type of configuration where instead of being concentric circles, you make spindle-like structure, which are also integer defect fitting with the poles of the sphere. But in this case, all the filaments are pointing towards the defect in a Naster configuration. There you get a deformation, and you very clearly see that the liposome is getting deformed by the moving microtubules and the force applied by this active nematic system. And this is essentially showing you and recapitulating everything that I'm going to tell you today about this active system. If you have a combination of order that can change with time, with forces that are coupled to the order you have in the system, there you can do punctual and predict shapes that will appear in the system. So this system, which is molecular-based, essentially uses all the ingredients I'm going to show you about at the tissue level. So what happens with cells? Because that's the big question of today. What happens with cells? So now you have active cell nematics, which are essentially cells that you put on a plate. And the cells I'm showing you here are called C2, C12. They are myoblasts. So they're very contractile cells, but also very elongated. So it's a perfect system to look at from a nematic point of view because they will form spontaneously at high density this nematic order that you see in green in the box. And again, in this nematic order, you have two kinds of defects, the +1/2 and the -1/2. Again, this is on a non-confined flat layer of substrate. So the +1/2, and let me explain you how we calculate the charge. So this +1/2 number, if you look at the orientation vector of the cells at one point around the defect, and you spin around the defect is shown as a red dot here. If you spin around the defect, you see that the vector field is actually spinning in the same direction, but makes half a turn when you do a full turn around the defect. So the plus says that the vector field is going in the same direction than the spinning direction that you apply. And the half makes half a turn when you do a full turn around the defect. And the -1/2 is the same. You see that you're spinning the other direction, so that's a minus, but you do half a turn when you do a full turn around the defect. So this charge number defines a certain type of orientation patterns around the defect, and with a defect of a certain charge, you get a certain type of orientation around the defect. So these are the essential ingredients that have been used by many theoreticians, but in particular our colleagues in Geneva, Carsten and Dan, to describe active stresses in active nematic systems. Why is that? It's because one of the things you need for morphogenesis is gradient of stresses. If you want to deform something, that means you apply more forces somewhere and that you create gradients of forces. And the nematics are very good at that because they have a certain order with defects. And so they have a gradient of organization, which if it's linked to active stresses, will create automatically, I would say, a gradient of stresses. So it's a very good way, if you wish, to describe those gradient formation in a biological system. So essentially, you have a contractile particle, which is essentially the microtubules or the contractile size, and you will organize them through a nematic order, and you will try to make sense of how the order plus the activity of the system creates different types of shape. I'll get a bit more quantitative into that. You do not need to know the details of the mathematics. This is the Frank free energy. It's the energy that describes a liquid crystal, a nematic in particular, and it has three terms. And the three terms are energy associated with the deformation of the nematic order. You can do splay, which means you don't have perfect order between neighbor particles, and in that case, it will cost you a little bit of energy. You can have a twist. This is in 3D. If you have a layer of nematic particles and you look at the other layers, you see that there's a change of orientation through a twist. And that's very common, for example, in cholesteric systems, and that will cost you energy. It doesn't exist in the 2D systems. And then the last one is the bend, where the particles in one line of order will not be perfectly aligned, and you have a change of direction through space, essentially. And with this, you can describe essentially all the energy associated with a liquid crystal. What you can do is actually couple this in the energy description with a surface deformation. And surface deformation, in particular in liquids, relies only on two terms. The bending term, which is the curvature associated with the bending rigidity. Kappa B is the bending rigidity, and h to the square is the curvature to the square. And you have, of course, the tension, which means changes of area in the surface. And what you can immediately see in that type of description is that you link through energy the orientation of the nematic order and the deformation. And just to give you a simple thinking about it, it's quite natural if you think of a tube or a cylinder that the preferred orientation will be along the axis of the tube in that case to minimize the energy because you will have minimal deformation of the orientation if the nematic order is along the axis of the tube. So even in passive system, you have a coupling between deformation of the surface and the orientation of the nematic order. But what happens in active systems? So I don't show the equation. These were the only equations I was planning to show you. But let's say you organize your particle into concentric circles. That's one test. And you can also organize your particles as asters, as you can see here. For sake of theory, you want to linearize this and make it continuous. So you have an analytic description by making just a field of orientation, either a vortex or an aster. Now, what you do is that you put these structures embedded into a flat surface with no active stress. And then you activate in silico the stresses. And what you see is that this flat surface change shape because when you put the stresses, you create force gradients associated with the nematic order, and you will deform the surface. But you see that you don't get the same shape as a function of the nematic order. These are exactly the same forces you put in. It's just the nematic order that is different. In one case, you get a saddle-like shape structure, and in the other case, you get essentially a dome. And just to mention, one is, of course, a negatively Gaussian curvature, and the other one is a positively Gaussian curvature structure. So that's exactly the kind of ideas we want to describe. Can we create shapes within flat tissues, initially flat tissues, by having an organization of cells and having active stresses? There's good hints in the literature about these ideas. I think one of the most famous work was done by the group of Benoît Ladoux, where they looked at the traction force microscopy around cells that were making +1/2 and -1/2 defects, as you can see here. And you see this is an average image of the stress field obtained by traction force microscopy for many different defects that were averaged together. And you see that you have a stress pattern that is associated and coherent with the orientation order of the cell, so the nematic order. And associated with these compressive stresses that they observe, they could show that there's a higher probability of extrusion of cells at the central point, which is at the defect site, because the stress field compresses the cells at the defect site and pushes them out of the plane of the layer, cell layer. Okay. a bit more complex now. The theory of active system essentially takes into account one type of forces, which can be contractility, but there's another type of active forces, which is motility. And I'm trying to distinguish motility from contractility. The main difference is that, at least for theoreticians, is that one is forces applied to the substrate, and the other one is forces, contractility forces applied to neighboring particles. So it's interaction between particles and the substrate in one case and between particles in the other case. What Carles did was to actually do a theory that was more complex, using two terms for active forces, so one term for motility and one term for contractility, and that allowed him to predict a full variety of stress field associated with the same nematic order. So if you see the three rows here, I should say the first column on the left is the Aster configuration. The second column on the right is the spiral configuration, and each row corresponds to different sets of parameters within the theory. And what you can see is that A and B corresponds to the same set of parameters, and it gives an extensile stress field. The arrows, the purple arrows are pointing out, and this corresponds to extensile stress fields, both in Aster and spiral configuration. The central row is another set of parameters, and you can see that we have a contractile Aster and we have an extensile spirals, whereas the last row, E and F, shows contractile spirals and Asters. So depending on the parameters you have in the theory, you can change completely the stress field and thereby the gradients of stresses and the expected shape as a function of the active stresses you have in, but also the nematic order you have. So what can we do with that? Well, we wanted a simple system to test this. And we got a little bit of inspiration from a very old paper. I mean, very old, it's 208, it's not so old. But it was done in plants, and that's the actually first system where people looked in details during morphogenesis, the orientation of cytoskeleton, cells, and shape that you get. And what they saw is a nice correlation between the orientation of cells, the orientation of the microtubules, or the cytoskeleton within the cells, and the overall shape of the stem, which is in this case at the tip of the stalk in Arabidopsis. And what they observed, if you look the panel D, is that at the tip you actually have no order. The cells are kind of oriented in every direction, whereas on the side they are all oriented circumferentially to the stalk. And when you create a new branch or a new stem, you see that you have a phase of disorganization and you have a proper order between the two stems, as it's shown on the drawings on the right. And that tells you that there's a clear correlation between the orientation of cells, the cytoskeleton, which produces the forces needed for morphogenesis, and the global shape that you get at the end. So that was one of the first systems shown here. So what Pau did in my group was to use a trick of confining the myoblast onto micropatterns by very standard techniques, where you use a surface coated with a repellent for cells, and you burn it through a laser at specific place to form discs of adhesion. And in those discs of adhesion, as you can see from the drawing and the picture below, on the left, you see that the cells initially organize into a spiral configuration. The cells are actually spinning. I'm going to show you this in a second in the disc. They keep on proliferating because it's a growing and it's a cell line, so it grows. And through proliferation, the configuration of the cells changes from a spiral configuration to an aster, which is also an integer defect somewhat. But this aster is more contractile, and so the cells are pushed in the middle of the aster configuration into a mount, and now we go from a 2D structure to a 3D structure. We create a multi-layering system, if you will, or a multi-layered system. And interestingly enough, the cells at the top of the mount will revert to a spiral configuration because the density is lower. And it's like if they had gone back to the initial stage of the spiral. So now you have a mount with a spiral configuration at the top and an aster at the bottom. And through proliferation, this spiral will transform into a vortex and grow into a tornado. And you can see even better by looking at each stage dynamically. You see that initially on the left, the cells organize into a spiral and rotate on the micropattern. The spiral at the top of the mount is also rotating, even though in that movie it has a opposite chirality if you look carefully. And then on the right, you see a full tornado. I'm going to show you a picture of these tornadoes. It can be several hundreds of micron long, and it rotates. If you look from the top, it rotates like a vortex or like a tornado. They can get very long, up to 0.5 millimeter, and they get this twisted shape that is also reminiscent of the rotation that they have all the time. So it's really a dynamic system, and we think it's a simple protrusion system or in vitro morphogenesis process, essentially. Now, what we wanted to know is how this is close to situation that you find actually in the real animals. And we wanted to know if the topological defects, which are the essential element of the nematic order that drives the morphogenesis in these cellular tornadoes, whether they could be found in embryos, and in this case, what could be their role there. So we turn to a very simple animal, which is the hydra. And this is the work of Yamini, and I'm going to show you a bit of this work. And you will see immediately the connection with what I've told you before. The connection is that in the hydra, the actin is organized as a beautiful nematic order, which spreads, tens of microns, so much bigger than the cell, and it essentially forms a layer of muscles that are connected through intercell digitations. What is interesting is that the hydra is a very simple animal with only 2 layers of cells. The external layer, which is the ectoderm, has a nematic order of actin filaments that is organized longitudinally to the animal axis. And the endoderm has a circumferential organization. So it's essentially like 2 muscles 90 degrees apart, and they beat in opposite in out of phase. They work like this. What is interesting is that the position of defects is not random in that animal. Actually, there's an aster configuration of the actin filaments at the mouth, which is at the top of the head. And there's also a nematic integer defect at the foot, so just on the opposite. And you can see that the line that connects these 2 defects is perfectly matching the axis of the animal. And of course, this is a very interesting observation. And so we wondered whether the fact that those topological defects were present in this position in the adult animal could have a role in forming the animal, which is essentially development. So the hydra, I should say, is very closely linked to Geneva, because the hydra was actually characterized as an animal by Abraham Tremblay, who was a prof in our university quite some years ago. And more than the person, you probably know this very famous experiment that he did, which is essentially taking a hydra and cutting it into 2 pieces and looking at the regeneration of the head from the foot part and regeneration of the foot from the head part. So you know that hydra is a very good model for regeneration and also for studying associated morphogenetic processes. So what we wanted to do in those experiments was to change the nematic order during regeneration to see whether it could trigger some changes to the morphogenetic processes. So the way Yamini did that was to actually use agar slabs to slightly compress the foot tissue, which normally regenerates a head. And in some cases, I'm not going to show you all the details. In some cases, looking under the microscope at what happens to the tissue while it regenerates under compression, she could observe the formation of two-headed animals, as you can see here. You also see the beautiful nematic order of the actin. These are life-act animal, meaning that they express a GFP tag life-act that labels filamentous actin. If you release those tissue that have regenerated two heads from compression and you let this thrive for 5 days, you actually regenerate fully viable animals with two heads and two parts of body, as you can see from here. You see that the heads have tentacles that can grab shrimps and eat normally, which is a physiological test. So what I'm telling you here is that in a normal developing tissue, you actually have two topological defects, which are the poles, and that one of the pole will become the head and the other one stays the foot. Now, if you compress by mechanism that we don't fully understand, we can generate a second defect on the side of the wound, which makes in total 3 defects. And in that case, you create 3 heads and 1 foot. So that suggests that it's absolutely needed to have a defect to generate a head, and that a bit more than this, it's actually a trigger. Having an actin defect is a trigger for the full program of regeneration of the head. Can we test that idea? So if you want to test the role of something in biology, the way you do it usually is that you remove it. You do a knockout, you do siRNA, you cut it off for development structures, and you see what's going on. Now, how to remove an actin defect? Well, we have a problem, and the problem is a strong problem. It's topology. On the sphere, you have 2 poles. So removing the defects from a sphere is essentially impossible because the topology of the sphere tells you that you will necessarily have 2 defects. So what we would like to do is actually make a torus, because on the torus, you have no defect, or you can have situation with no defects, I should say. Why is that? Well, let's go back a bit to the mathematics. It's very simple mathematics in that case, and it's linked to Gaussian curvature and topology. So on a surface, at any point, you can define different types of curvature. And these curvature will be linked to the radii of 2 90-degree apart tangential spheres, or I should say circles, at that particular point. So the Gaussian curvature is defined by this. It's 1 over R1 times 1 over R2. And if you integrate the Gaussian curvature over a sphere, you find a constant, which is 4 pi, which I can rewrite 2 pi times E, where E is actually the Euler characteristics. It's a number that defines the topology of a surface. There's a very complex theorem called the Gauss-Bonnet theorem that actually shows that on closed surfaces, this is a constant, whatever shape you get. Essentially, what it tells you is that when you have a closed sphere and you deform it, you won't change the topology unless you make a hole into it or you open it in some positions. And why the Euler characteristic is 2 for a sphere is because you have 2 poles. So you see that the number of defects is actually the number you get from the Euler characteristics. So now let's look what's happening when you change the topology of a sphere by either fusion or fission event. And depending on the way you do the fusion and the fission, you can actually increase the Euler characteristic or reduce the Euler characteristic. So I'm going to focus on the case where you reduce it. If you actually fuse the sphere inside, what you get is a torus, and the torus actually has a Euler characteristic of 0, which means that in theory, or in some cases, the torus can have no defect at all. And that's why we wanted to do a hydra as a torus, because that was the only way we had to actually remove the actin defects and test their role. And the expectation of the experiment would be that if we can get a torus without defects, we would get no head and no full regeneration. How do we do this? Well, actually, we were very lucky on this, is that the foot tissue, in some cases, instead of being compressed laterally, gets compressed along its axis. And in that case, you get these beautiful images where you are looking at the foot of the tissue and you see the aster configuration of the actin all over around the tissue. And so the center is the defect and you see the aster there. But it's compressed towards us. Essentially, you're looking with the slab like this. The tissue is trying to regenerate, and for doing so, it has to contract and constantly moves. And doing so, it breaks in the center, as you can see, and creates two holes that fuses back together, creating a perfect torus. Again, this is perfectly spontaneous under compression. It's just a matter of how the orientation of the tissue is under compression. And in this case, these tissue do not regenerate a head or a foot. This is a tissue that has been released after 4 days of compression, has been released, and we look at it for 3 more days, and you see that no head and no foot is forming. The tissue dies after a while, I have to say. Of course, it has no mouth, so it cannot feed on anything. So we were very happy with that result, because it means that if you have no defects in actin, you actually don't regenerate a head, which strongly support the idea that you need an actin defect to generate the full program of differentiation. However, we still did a major change. We changed the topology of the animal, which is that we transform the sphere into a torus. And maybe this is actually causing so many problems to the biology that it creates that's the topological change that actually triggers the absence of regeneration. So what we wanted to do was to do a torus, but with defects. And this experiment is interesting because that's the real test to whether the defects is mandatory for creating the head. Because if you have a torus with defects and you regenerate a head, that means it's really the defect that controls the head regeneration. Otherwise, you will know that the topology is controlling this. So for doing that experiment, we changed a little bit our protocol. Instead of using the head regeneration from the foot tissue, we used the spheroid. And the spheroid is the central part of the animal where we cut the head off and the foot. And it closes back onto itself, but because it has 2 wounds, it loses quite a bit of its nematic organization during the process. And you see this here. If you take a spheroid under compression, you see that there's a large part in the center at 6 hours post-dissection where there's no order of actin filaments. You still see the nice nematic order on the boundary of the tissue. And through time, this order will rebuild. But when it rebuilds, sometimes it creates a defect. That's the red row you see on the right panel. At the meantime, and for the same reason than the movie I've shown you before, it can break up and form a torus. And that's what you see on the final panel at 53 hours. You see that there's a hole, meaning that you have a torus, and at the same time, you have a defect. Now, if you let them grow and release them from compression, you get those animals. And these animals are viable. They have a head and tentacles, but they have a hole in the belly, which means you fully regenerate a head and a foot. But of course, you keep a strange topology because you were formed like this. So essentially, what I've shown you is a fairly straightforward conclusion. In the normal case, we have 2 defects, and we create 1 head and 1 foot during regeneration. If by compression we generate an additional defect, we get 2 heads and 1 foot. Again, through compression, if we generate a torus without defect, we get no head or foot regeneration. But if we can make a torus with defect, in that case, we create a head and a foot, but the animal is intrinsically a torus. So we think this shows that the topological defect in the actin order are required for head regeneration. And the concept behind this idea is essentially that the organization of the actin is there to shape the tissue into an elongated structure, an ellipsoid, to actually create the initial axis of the animal. So it's a very basic process, but very similar finally to the tornadoes I've shown you, where the protrusion and the axis of protrusion of our myoblasts is organized through the defects that is initially formed from confinement. I see that I'm already quite advanced, so I'm not going to talk much about the work of Claire, because this is very physics. It goes deep into controlling the tornadoes in vitro. Just to give you an idea of this, the question of the project is how the geometry and the topology of closed surfaces can control really morphogenesis. And in particular, we wanted to redo this experiment of tornado formation on the topology of closed surfaces. And I'm going to skip a lot of slides that I've here, and I'm just going to show you the concluding slide that we had. Yes. It's going to show the whole animation. I'm sorry for this. So what we think is happening on the closed bead surfaces is that initially we form +1/2 defect. Again, these are myoblasts that are bound to an alginate hydrogel sphere, so they are confined by the bead essentially. So initially they form +1/2 defect, like on the flat surface, but they grow multi-layer that I had no time to show you, and eventually reach a stage where they actually form +1 defects, so asters, as you can see here. And these asters will grow into mounts, as we had seen on the flat surface initially, but these mounts are unstable. They are not staying as opposite poles. They actually merge together to form a tornado, and this tornado is actually a +2 defect. So it's another a type of defect. It's one higher class of defects. And again, this is, I would say, speculative at the moment. We are looking into the different stages and look at the nematic organization of the cells and their dynamics, associated dynamics at different stages. So we can describe in very much details the different types of defect, the different dynamics of tissues we get, and the associated stress fields that shapes the tissue. This is interesting because we can control the charge and the position of the topological defects with the topology of the sphere and the shape. I didn't show you, but there we have tiers and spheres. We can show that in multi-layered system, it seems that at least the myoblasts prefer to organize as orthonematics, which means layers of nematic order at 90 degrees. So one layer in one direction and the next layer with a 90-degree order. And the combined effect of these increasing nematic order with time and the movement of defect associated with the active forces in the nematic order creates the apparition of +1 defects that grow into mount and further into a protrusion. The nice thing about this work is that with very simple system, we are able to reproduce what happens in many embryos, which is to go from a sphere configuration to an elongated protrusion thing by controlling the organization of cells at the surface. Okay. Within 2 minutes, I want still to show you this because we went a bit further into trying to reproduce in vitro the real morphogenesis. And for this, we used gastroloids. That's the work of Oriol. And this is very preliminary, so there's not a lot of things to show you. I'm not going to go into the details of gastroloids, but maybe you know what they are. These are made from embryonic stem cells. And if you give an induction of Wnt signaling that you stop at after 72 hours, you can induce the differentiation program and the elongation of the spheroids that are formed with these ESCs. And they look like this through time. They grow and they elongate. And so we wanted to understand what is the elongation process and whether the elongation process that is seen in gastroloids has nothing to do or has anything to do with the tornadoes we have been studying before. What is interesting is that if you put a membrane marker at day 3, which is right after induction of Wnt signaling or the end of the Wnt signaling treatment, you see that the cells are quite round shape and there's no particular order. But through time and during the elongation process, you see that the cells are getting more elongated and they seem to organize as a nematic order. These are many of these gastrolids at day 5, and you can see that they somewhat present a nematic order everywhere. And what you can also see is that it seems that the nematic order from these pictures is somewhat perpendicular to the axis of the growth. This is probably more easy to see, and you can, of course, quantify it by doing two things, essentially segmenting the cells. And that shows that the aspect ratio of cells is increasing with time, showing that the cells go from round shape to elongated. And you can also look at the orientation of the cells, and you can see that, sorry, I went a bit fast. You can see that the number of cells with a very oriented direction, so the red ones, is going up with time. The proportion of red cells is going up with time. We can do a bit better than this. We can look at the surface of cells. This is not, this is the same than before. We can look at the surface of cells and look at the orientation of cells at the surface of those gastroloids with a more complex analysis developed by Kostas in the lab. And what you see is actually that there's on elongated gastroid, you have almost a perfect order between the two poles, but you lose the order at the poles, which are the red regions that you see on this analysis. And these are where you have defects. So the defects are found at the pole, and the rest of the order is essentially organized perpendicularly to the axis. And that's support the same kind of ideas than what I've shown you for the tornadoes and things. We can quantify this in a pretty clear manner along the axis. And essentially what it shows, if you look the bottom row, is that the orientation angle it becomes very, very, the distribution of angles of orientation becomes very narrow when the gastroloids are elongating. That's the bottom row. What we wanted to see is whether this is associated with a drop of tension in the membrane. And the idea is very simple and shown by many papers, including Henri de Belly, who gave a talk in your seminar series a few weeks or months back, where during the differentiation of cells, you can get a drop of tension that is associated with a change of shape of the stem cells during differentiation. So we tested this. This is work from Tithi in the lab. And Tithi used a membrane tension probe that we developed a few years back with our colleagues of organic chemistry, Stefan Mathil here. And what she showed is that the tension is actually going down. The lifetime of the probe is going down, which is a signature of a drop in tension. And so what we think happens is when we, in the gastroloids, forgot to show the gastroloids. These are the gastroloids. You see that the lifetime goes down. That shows that during the differentiation process of gastroloids, the tension of cells is going down. So what we think, and that's the very last one, what we think happens is that the round cells that are pluripotent stem cells, when you induce the differentiation, see a drop of membrane tension and gets this elongated cell shape. And because they are elongated now, then they can create a nematic order because they're all packed together, and so they will align locally. And this nematic order feeds back onto the morphogenesis and controls the way the cells will organize into morphogenesis. We think there's essentially two hypotheses of how the nematic order will drive elongation. One is by T1 transition and intercalation of cells that are ordered by the nematic order, or the direction of intercalation will be driven by the nematic order, or by forming tornadoes. This is something we would like to test by making dynamic observation of the growth. And we think it could be a protrusion by a nematic order that rotates the cells all together and makes the protrusion in the same way than the protrusion. I'll stop here and thank the people, Oriol, Tithi, Claire, Pau, who was the postdoc and is now back in Spain, and Yamini. And with this, I'll be happy to take questions.
Great. Thanks a lot, Aurelia. So for the questions, we do as always, please write your questions in the chat. And while we wait for them to roll in, I will start. So I was wondering, how important are the cell intrinsic? You got to this in the very end a little bit.
Yeah.
What cellular features are needed to establish such a nematic order? Like you said, the loss of tension allows this, but I mean stiffness or viscosity or these types of things.
So you can have several types of nematic order. The most classic one is what we call the orientational order, which is associated with the cell shape. So what you need is an elongated cell shape. And if a system that has where cells are elongated means usually that the tension of the cell is pretty low. Otherwise, it's a bit like a balloon and it keeps a round shape, and there it becomes less nematic intrinsically. So we do think that controlling the membrane tension at the level of the tissue allows for the formation of nematic order. We have, as you have seen from the gastroloids, we have a sort of hint about this happening there, but it could be a more general process where tissue regulate membrane tension of different types of cells and create nematic order in order to create different types of shape associated with the differentiation pathway chosen by the cell. So that's one option. What you need is active stresses. So you need the contractile system. And I always find funny when you talk with people doing organoids or gastroloids that they always have to treat the cells a little bit with actin drugs to lower contractility in order to induce differentiation, but also induce the correct pattern of morphogenesis. So I find this interesting that there's a link between contractility, shape of the cell, and then the morphogenetic process behind.
Yeah. Okay. Thanks. So Yuping is next.
Hi, Aurelian. That's beautiful talk. I have two
Thank you.
questions. First question is, can you explain how pressure from the agarose pad translate into the topological defects in the actin filaments? That's for the first question. And my second question is, both the gut of an animal and the torus you've shown have the same topology. How are they developed differently?
Yes.
Thanks.
Can you repeat the first question, please? Because I didn't hear the beginning.
Right. My first question is how does the pressure from an agarose pad translates into topological defects in the actin filaments on those hydra tissues you use?
Yeah, that's a very good question. What we think happens is that in the normal case, actually we use a dissected tissue. So essentially, we cut the animal in two parts, but we inherit, the tissue we use inherit the nematic order from the foot of the mother. So we have something like this. But then, of course, the tissue has to heal. So it closes back by closing the wound. And when you close the wound in the normal case, you recreate an Aster defect because the nematic order you inherit from the mother will close back at the position of where the head was before dissection. Now, what we think happens is when you compress with the agar slab, what happens is that instead of closing isotropically, you actually close anisotropically because the tissue is compressed, and so you have a line. The wound is a line instead of being a point, and this line somehow breaks into two defects. This is something we don't really understand in details, but we think the compression, because it's an anisotropic compression, allows us to split one wound into two, essentially, and that's why we get two defects at the end. That's the answer to the first question. And the second question was about the torus, right?
Right.
And it was, can you repeat it?
Oh, right. It's how is gut from different from a
Yeah,
torus?
that's a very good question. So, as you all know, we have gastrulation, so we have a gut, and the question that you can ask is whether we are not intrinsically torus, because at the end of the day we have a gut, and so we could be seen as torus. First, for the case of the hydra, it's a bit different because the hydra, most of the time the mouth is closed. The mouth opens actually like a tear in the tissue. So the hydra is intrinsically a real sphere. So this is a bit of a difference between hydra and the other types of animals. And second, at the end, I think what the study shows is that it's not really the topology of the tissue that controls anything. It's more the defects that you have in the tissue, at least the organization of the nematic order in the tissue that controls the orientation. In that case, you can have the same processes with a completely different topology, whether you are a torus or a sphere, essentially. And actually, we have a hydra with a torus, but still regenerates a head and a foot. So my simple answer, it would be that it doesn't, at the end, care very much whether you are a torus or a sphere.
Okay, gotcha. Thank you.
So then, next question. Thank you.
Hello. Hey, Orlin. Thanks
Hello.
for the really nice talk. I'm
Thank
just
you.
wondering about the last piece of your data where you showed
Yep.
this decrease in membrane tension during differentiation. I wonder how much contribution there might be from lipid composition changes during differentiation process.
You are totally right. We think there's actually quite a bit of change in the lipid composition depending on differentiation. There's actually more and more lipidomics analysis of the membranes of stem cells going through differentiation. And of course, this could affect the response of the probe that we are looking at, which is this flippler. However, there is still a shape change of the cells. They go from round shape to elongated, and this should be associated with a reduction of membrane tension to some extent, or an increase.
Why is that?
Because if you have a very high tension, you will tend to be round shape, whereas if you have a lower membrane tension, you should be able to be more easily deformed, unless your contractility goes up.
Right.
And it could actually be a combination of both, which is a drop of tension and an increase of contractility in order to be able to form elongated cell shapes. But it's a fair remark, and it's a fair point.
Right. I was wondering if it's possible to sort of calibrate the tension probe under different stage of the development, and then, well, under different type of cells. I'm not sure. So that the lifetime really tells you potential tension changes.
Yeah. So in a single cell line, you can do that. So we have done it for HeLa cells, for example, which is to calibrate the lifetime as a function of the tension, which allows you to somewhat measure directly membrane tension through the lifetime of the probe. However, you have to have a calibration for each cell type. We actually did it for two cell types, MDCK and HeLa, and we see that the curves are very different from one to another. So it's indeed very dependent on the cell type, and that's for a simple reason that you actually very well said, which is that the lipid composition is different, and the lipid composition affects the response of the probe.
Right. Okay. Sounds
So
good.
So that could be a thing. One thing I want to say is that we are trying to use this probe to see gradients of tension in tissues.
Right.
And that is something we start to see. And of
Mm-hmm.
course, these are tissues made of homogeneous cells. So that means the
Mm-hmm.
cells are all the same. So the
Right.
difference cannot come from a
Right.
lipid difference.
That'll be cool. Yeah. Thank you.
You're welcome.
Mm.
And David.
Hi, very cool talk. Thanks. I was wondering
Hi.
about the topologies. How stable are the topologies? If you already have one topology, can you change back to another one? Or is it already like you're committed to that one and it would continue growing in that direction?
You mean the topology in the tissue? If you have
Yeah,
if you are a sphere or a torus?
right.
Yeah, that's a good point. What we see is that the change of topology occurs pretty early after regeneration, and it may be associated with the fact that the nematic order of actin is not very well specified at the beginning. So it seems that you can change topology in a tissue that is essentially repairing or regenerating. But once you have done this, the tissue seems to be fixed in its topology and doesn't change much. And you have seen it from the fact that we get fully developed animals, and they have not changed their topology even 5 days after release from compression. So they usually change their topology, acquire a certain topology in day 1 or 2, and then they keep it for the rest, even after release from compression. That's what I can say. But in theory, it is possible. Nothing says that if we take those animals and cut them back into pieces and compress them again, they will not change back to sphere topology. This we have not tried, of course, but there's no reason why it would not be the case.
Okay, thanks.
Thank you.
Tomoko.
Hi, thank you so much for the beautiful talk. I think my question is somewhat related to David's questions. I'm curious how robust is the thematic order of the biological systems. So, for example, for in Hydra's case, a continuous perturbation was used. But is it also possible just like one-time perturbation, like maybe steering or making a hole or something could potentially change their order and they affect the defect number or
Yeah.
the type?
So yeah, there's plenty of very good questions. There's plenty of things to say about your questions. The first thing is how stable the nematic order is strongly depends on what type of material you're looking at. If you're a solid, the nematic order is fixed where it is because you have a solid and you can stretch, you can expand, but the position of the particles and the way they are oriented will not change much through these elastic deformations. That's the case for the Hydra. We think the Hydra is a solid elastic, and that's why through compression we change a little bit the order, but not too much. And that's why it has such an impact. But this is not a classic description. Most of the people that describe active nematics, they use a liquid description where the particles can, slide and move and change position. And in that case, the order can change dramatically if you change, if you deform the volume or if you make flows. And so this is something that is becoming a subject of interest is how much the nematic order of cells is linked to the phase property of the tissue, whether it's a liquid tissue or a solid tissue. And you know that in development, there's more and more reports about the fact that you have transition from liquid or phase transition between liquid and solid tissues at different stages of development. So that there could be a link between the capacity to change the nematic order when you're a liquid and to fix it for a while, at least during a morphogenetic process, because now you're a solid tissue. That's the first thing I wanted to say. The other thing about the local change of nematic order, one thing we would love to do in the Hydra would be to do laser ablation, like a punctual hit to the tissue, and see whether we can locally reorganize the nematic order and see whether this is sufficient to create a head. With no, we don't cut the animal at all. We just make a little hole, and maybe there we can reorganize the nematic order a little bit and see whether we can create a head in a pre-existing animal. This is a bit of Frankenstein experiment, but we didn't try.
I see. This sounds very cool. Yeah, thank you so much.
I have another question along
Yes.
these lines, which is a little bit, what's upstream and downstream, because in your latter part, right, you induce the differentiation with Wnt and you change the cell properties and they elongate, and then that can form the order. But in the Hydra, you're preventing the formation of the defects and then you're not differentiating. So is there, like, do you know, I don't know what kind of transcriptional
Yeah.
changes happen in Hydra differentiation? Are those all suppressed in that case?
Yeah. So you're asking an essential question here that I often get from the biologists, because in biology, where you have the head is actually where you have the Spemann and Mangold organizer, which secretes Wnt. And the idea of how it creates the symmetry breaking that leads to the axis is essentially a Turing mechanism. You have a source that secretes a diffusible gradient, and the gradient is actually formed by the fact that you have an inhibitor that diffuses much faster than the diffusible morphogen. The problem in Hydra is that they have the morphogen. It's Wnt3. It's very well established, described, and it's a general thing. It's not just in the Hydra, it's in every animal. But they never found the inhibitor of the Turing. So it's a frustrated Turing process where you have one of the two parts of the classic Turing patterning process. Not just our work, but some of the work from other groups have pointed the fact that the mechanics may be the inhibitor, and that maybe there's a direct coupling between the way the forces are generated in the tissue with the secretion. There's work from Kinneret Keren and also Karisios Tsaris in Basel that have shown that there's a link between secretion of Wnt, the nematic order, and the stretch linked to the nematic order. And why is that interesting? Because now it somehow puts two things at the same level. Before, people thought the Spemann organizer dictates everything, the transcriptional program to differentiation, but also the forces needed for forming the tissue. What our work suggests is that actually the forces, the mechanics, and the transcriptional control by Wnt are at the same level, and they are part of a feedback loop that can induce. So you can induce the differentiation program by putting more Wnt, or you can induce the differentiation program by putting more mechanics and active tissue. So that re-equilibrates the role of mechanics, I would say, with the transcriptional control. So it's not shown the thing, but very interesting problems. That's where we're going.
I mean, your donut is suggesting that this is happening, right? I mean,
yes, exactly.
There's good evidence for that, right?
Exactly. The fact that you can completely block the regeneration program by removing the actin defects is almost a direct proof that it's at the same level than the Spemann and Mangold organizer. Yes, that's true.
Well, really cool.
Very cool. Thank you.
Okay. So if there are no more questions, then thanks, Aurelia, for being here. And
I was very happy.
I wish you all.
And thanks again for the great questions.
It's really nice. Thanks. And I'll see you guys next week, or 2 weeks, in 2 weeks. Yeah. Okay.
Bye-bye.
Bye. Thanks.
Thanks.
Bye.
Thank you so much.
Bye-bye.
See you. Bye.
Bye-bye.